heard about Paul and John's work. No, sorry, I'll let me just say a little bit first, actually. I made some hybrid presentation, a little bit of this, a little bit of that, a little bit of that. But my general, what I'm working in my research is trying to understand the role of stochasticity in biological function. And I'm working on 3 model systems: immune signaling, chemoreception, and development. And across these systems, there's something ubiquitous we see is that when we look at the back of the watch, take the watch face off, we see all sorts of chaotic signaling apparatus going on in the background, yet on the front, things appear to be working well deterministically. And I know the theme of the conference is cancer, which is obviously when things go wrong. But as was mentioned by one of the speakers previously, the vast majority of tumors are identified by the immune system and eliminated, and it's something that goes wrong after a long period of time. So one of the things I'm working on in my research is how to characterize this deterministic outcomes we see in biological systems, and not in terms just of the robustness to variability and biological variability and stochasticity, but actually a constructive role that stochasticity can have in enabling the correct function. But now I'll talk about my postdoc research, which was on something completely different, which was on essentially micro electromechanical systems, and it was about modeling how membranes work when they're actuated by electric forces. And engineers were working on these a lot of the time because they'd learned to shrink things down and were building microscopic machinery. And as you shrink things down, the ratio of surface areas to volumes becomes such that you want to use electric fields. So I was building mathematical models to understand how elastic structures deflect in the presence of electric fields. And these are the sort of equations I would write down. It's actually similar to a lot of the diffusion equations we're seeing, except that in place of diffusion, there's an elastic restoring force. In the end, we'd get all these nonlinear systems of equations. And I was doing numerics on them. They would have singularities. They would have sharp interfaces. I spent a lot of time doing moving mesh methods for nonlinear PDEs, where the PDE would get some singularities. Then the singularities would kind of stabilize and evolve. Build a lot of numerical methods for how to resolve these accurately. And we would derive these motion law equations for the effective dynamics of these interfaces. And there were mean curvature equations. And of course, as I was saying to many of you before, I would derive these things, and my numerical approach is always do the simplest thing first. So I would write down some curve in polar coordinates, and I would calculate the curvature, and then I would try and solve equations like this with like forward Euler. And I just never met something that was so unstable in my life. And of course here, rho is the normal vector to some curve. So you have some curve here, and you can write down a shape. And then of course, in terms of some polar coordinate, you can write down the curvature and try and do simple things. And it doesn't work too well. And I don't know if I'm ever going to be able to solve this. Why is this so difficult? And it was actually reading Paul and John's papers that I kind of learned that, yeah, there's a lot of people who've been working on this level set method. You don't simulate this thing directly. You embed it in this higher dimensional setting, and I was able to get this working. So thank you for all those wonderful papers you wrote a long time ago, which I needed to sort of get my own papers out when I was a postdoc. So I appreciate that. And so let me talk about what an extreme statistic is through an example. So this is an example of moth mating. And the situation is there's this red dot on the left is a female moth, and they go through these calling signals. So they emit a plume of pheromones. That's what these contour lines here are. And then there's some wind, and here you can perhaps see that the wind is going from left to right. And then the male moths eventually detect this, and then they undergo a kind of well-known pattern of events. They go for a random walk, and then whenever they hit the pheromone, they surge up. And if they lose the pheromone, they'll try and cast to get back, and then eventually they'll find the female. And as you can see in this simulation here, there's one of them have met, two of them have met, and it's the first one that gets there that has the chance to mate with the female. It's a similar problem in fertilization where there's one egg, but millions of sperm that are trying to find that egg. And the point of this, this model we built here was that if you're interested in looking at the mating time, and I'm trying to understand, and the point of this paper was to understand how different calling strategies, different pheromone plumes would influence mating outcomes. Who cares what the average time to get to the female is? All that matters is who gets there first. Similar situation in fertilization where there's millions of sperm competing to fertilize one egg. Who cares what the average time is? All that matters is who gets there first, which sperm gets there first. And that's an example of an extreme statistic. So let me describe a little bit the mathematics of extreme statistics. So suppose we have a whole bunch of random variables, and these t's here you can think about just being the arrival time of any of those moths. And if they've got some PDF f and some survival probability s, then we can write down the distribution of the minimum. Okay, it's a sort of elementary statistic here. And if I plot this for a characteristic PDF, so if I take a PDF here, and this is a PDF that you see all the time in diffusion problems, t to the p, e to the -1/q, it's coming from the heat kernel. It comes up all the time in the diffusion problems. And it's very wide. It's saying that for one incidence of this stochastic process can have a very, very wide range of outcomes. But as we increase the number from 1 to 20 to 100, things become very highly focused around defined mean with a tight variance. So extreme statistics have this capability to give something extremely reliable from something that has a very wide and unreliable, non-robust outcome. So let me talk about, that's one example, the moth mating. Let me talk about two examples that have come up as well. Let me talk about how this can be useful in immune signaling. So there's a few people who've talked about a little bit, there's a bit of introduction here, and I've also worked with Omer at Oxford. Well, T cells have to make a decision. They have to make a decision whether cells they encounter are hostile or not. Should they be attacked or should they be left alone? And they interrogate the surface with their T cell receptors, and it takes them about a minute. So they come up against an opposing cell, they smoosh into it for 1 minute, and then they make a decision, attack or move on to the next cell. And anti-discrimination is the property that T cells have is that they can make that decision pretty quickly, and that they can tell whether there's low affinity antigen that come from proteins expressed naturally by our body, or higher affinity non-self antigen that are indicative of a pathogen. And this particular project was motivated by some advance in microscopy that said, look, these aren't two spheres that smush each other, they're two hairy balls that smush each other. And the surface were covered in these little pokey rods called microvilli. And instead of really just smooshing into each other, the T cell really just stabs the opposing cell. Now it stabs in such a way that you can see here, this blue curve is how long it takes to stab the whole surface. It takes about 1 minute. But while it's stabbing it, it's only stabbing at 40% of the surface at any one time. And we want to kind of understand why that was. So we built a stochastic model of this, an agent-based model, where this interface was a circular region. There was these microvilli tips, which are rich in TCRs. And it's like a search problem. They're trying to find these little dots on there that are the proteins they want to interrogate. And as Jun mentioned in his talk, there's a sort of well-known model called kinetic proofreading, Keithan's kinetic proofreading, to try and understand how TCRs engage with antigen and decide whether a productive immunological response is warranted. So they bind to them, and then they can undergo a series of transformational steps, phosphorylations, and then once a finite number of those is completed, a signal can emerge. All the while, they're subject to dissociation. So it's climbing a ladder. You have to climb all the rungs of the ladder, but at any time, you could just fall off and lose all your progress. So we built this model, and this is what it looks like. So this is the interface between those two cells, and there's all these microvilli. They get dropped in automatically, randomly. Maybe they land on a spot where there's an antigen that needs interrogation. Maybe they don't. And if they do, they can try and engage with it through this kinetic proofreading mechanism. And this was calibrated, and it ended up agreeing well with the data. And what we found is this 40% actually just comes from geometric packing, in the sense that if you try and smoosh two hairy balls in together, then they can only really get 40% contact area, because to do better than 40%, you need non-random packing. And so if you try and fill a disk with other disks, you get to about 40% if you pack it random. Now, this is at steady states. You see all these, the color indicates that that whole kinetic proofreading path has been completed, and they're signaling. But really, if we start this again, if we start this again, then really it's that first signal that should be the one that's most informative. So it's the first time that this gets activated, is really when the cell should make its decision about, there's an argument to make that this first one, which is much sooner than it takes to evaluate the whole surface, this first signal here is what you should sort of make your decision on and respond on. So we went back and made a classification model that did that. Like, what would you get the first signal we get? We're not going to wait till steady state. We're not going to sit there for a minute and just interrogate the whole surface and see what we get. First time we get a signal, we're going to make a decision on this. That's an example of an extreme statistic. So each one of these are an independent random process. We're going to take the first one of that. And when we did that, what we found is that we got something that massively improved traditional bounds on kinetic proofreading. And we got a curve that looked like the following, in that as a function of contact time, it gives you an optimal time you want to be in contact with the other cell. And if you wait too long, you're going to make more mistakes. So if there's a very long contact time, you have a very low accuracy in your classification because you're making false positives all the time. Whereas if you have a very short interaction time, you might have low accuracy because you're making a lot of false negatives. And of course, there's sort of an argument that it's better to make false negatives than it is false positives in an immunological scenario. And this kind of gives an explanation for why we often see very short transient contacts between T cells and APCs before eventual long contact when there's a signal is being received. So that was an example of extreme statistics in immune signaling. Another example in chemoreception, and this is some experiments that have been observed in this particular data set's over 20 years old now, but it's an example of neutrophils. And of course, we've all seen that famous video where the neutrophils following the bacteria. And essentially what happens is when a pipette with a chemotractant is used to elicit a response from these neutrophils, it happens very, very quickly. So in these experiments, they just pipetted a tiny little bit of a chemotractant to the left of this cell here. It's a little bit difficult to see with the contrast in the lights, but within a second or two, the cell knows where to go and moves to the left towards the source. And there's other experiments where you can play games and confuse the cell. So it starts moving to the left, and then you drop things over here, and it gets confused and tries to move to the right. And you can play all sorts of games confusing the decision-making process of the cell. But the basic thing is here, there's some source here, there's some diffusing molecules, and the cell's got to make a decision where to go based on engagement of those molecules with its surface receptors. And in various different types of cells, there might be clustering. So there's some initial signal, and then the receptors cluster in the surface towards that signal. But there's an eventual movement towards the source. And so we want to understand the certain timescales and how the cell can get the information it needs to decide where the source is. And this is a diffusion problem. So it was interesting to hear about these other structures that cells can use, long directed, these microtubules that cells can use to communicate with each other. Here, we're just assuming pure diffusion. So there's some cell here, it's got to react to this source here. How can it infer the direction through activity at its receptors? Now, there's various assumptions here that I'll just go through quickly. It's a pure diffusion here. There's some delta function initial condition to the right here. We can see the answer. The cell just gets the fluxes and it has to infer the initial condition from the fluxes over the boundary. Now, if we do this in 2D, then of course we know the random walks are recurrent. That means that every single one of these green balls will hit one of the receptors. In 3D, they can go off to infinity, but in 2D, they'll each get to one of these baskets. And this idea of ratiometric sensing is that, look, if the source is to the right, then more will engage at the front and then they will at the back. Because if the source is right there, there's going to be more engagement to the front receptors in the back, And through that, the difference between receptor activity at the front and back, we can make a decision where the source is. Now, it takes a while for all these diffusing molecules to get to where they're meant to go. They'll all get there eventually, but it can take a long time. And in the meantime, the cells should have to store and compare all those activity levels. So they need to have some memory, and then they have to be able to integrate that signal. So they have to store the signal, the memory of it, and then they have to integrate it. So those are just things that the cellular computational capacity that ratiometric sensing involves. And as other talks have mentioned, we want the cell has to solve an inverse problem. It needs to know the source, but it has the solution of the PDE. So it has the solution of the PDE. It wants the initial conditions. It has the flux over the boundary. It wants the initial conditions. So we use a maximum likelihood approach to do that. Of course, there's this argument, can the cell do MLE? There's arguments about, yes, it can. But we used a maximum likelihood estimator to build a likelihood landscape for where the source is. And when we look at this, so here's an example of what we saw here. So there's surface receptors. And if you wait for all the particles to get absorbed, you get a pretty good, the cell can recover pretty well where the source is. And in particular, it does a really good job on the on both the position, both the distance and the angle of where it is. So I'm kind of reading from right to left here, whereas on the right, you've let all the particles arrive at the cell, and you've built an estimate on where the source is here. But surprisingly, as we decrease the time and just look at the earlier and earlier arrivals, you see that the directional estimate is really good, but the distance estimate is worse. So by using fewer and fewer a smaller and smaller portion of the signal, the cell can improve their directional estimate, but at the same time deteriorating their distance estimate. So the short time signal here yields the direction of the source. And we can take this to an extreme statistic scenario as well. So here's an experiment. The source is over there to the right. So for example, here I put it at 5, 0. And then it's in polar coordinates in 2D. So you can see theta is the arrival angle. So we can think all those particles are going to arrive on the surface. And then we can order their arrival times. And we can say, okay, suppose I just take the first one that comes, the first arrival, and I just go in that direction. Well, what happens if I do that? Well, what we see is that if we take a bunch of them, and if we average over the angle that's coming in, what you see is for quite a long time, the estimate is really good. So the true answer is obviously 0. So for a long time, if I just take the angles that are coming in, then it's actually a really good estimate of what's happening on the surface, where the source is. And then at some critical point, it looks like roughly t to the time is 10 to the 2 here, the estimate decreases, and the KS statistic is comparing with a uniform distribution here. So at that time, everything is essentially uniform. So at short arrival times, there's a very high quality estimate on the directionality of the source, which deteriorates at some fixed time very rapidly. And we can analyze this using a boundary homogenization technique. So boundary homogenization is this way of replacing receptors all over the surface with some effective porosity condition. So you can change all that Dirichlet-Neumann and replace it with a single Robin condition that mimics the flux of the total arrangement. So if there's circular holes on the edge of the membrane, we can divide them up by keeping the area, the total absorbing fraction fixed. And if we divide them up, we see that we get something radially symmetric. And we can get the effective flux here and replace those Neumann-Dirichlet boundary conditions with an effective Robin condition. That allows us to do some analysis. And what we see here is that if we just do a separable solution, there's two curves here of interest. The blue one is basically the first mode in the Fourier series. It decreases rapidly as a function of time. Whereas the 0th mode, which is the CDF, of course, it increases from 0 to 1. And what we see is that very rapidly, the solution loses radial symmetry, indicating that quickly all the particles that either get to the cell very quickly and have the directional information. They either get there quickly or they diffuse around and lose all the information about where they started from. So if we take this to the extreme, so I go back to that experiment again, take a large number of source particles and just say, all right, what's the error in moving in the first direction? I take the very first particle that comes to me. What sort of error do I make if I do that? And the answer is it's normal. It's normally distributed. And the error is the variance in this estimate is this function here, where r is the distance of the source. And it decreases like 1 over r. Obviously, when r is close to 1, when the particle is very close to the source, it's a very good estimate. And when the particle is very far away, it decreases, and it decreases like 1 over r. So that shows that a source that's twice as far will be twice as difficult to locate, or the error will be twice as much. And we can do this in, well, sorry, if we get some numbers from these experiments, and this is just sort of diffusivity, the sort of concentration they used, we can get that the time is about 1 second. So for the first particle to arrive is roughly 1 second. And in these experiments, they observed a cellular reaction in about 2 to 3 seconds. So it gives a minimum theoretical time for a cell to respond to a diffusive signal. Of course, that particle arriving at the receptor just initiates some downstream activity. So it's truly is just a minimum theoretical bound. And we could do the same thing in 3D as well. So that was in 2D. In 3D, you see that the elevation coordinate is exponentially distributed with a very similar error. So the dimensionality is not affected much. In fact, we just get this constant, this factor of 2 in the denominator there. So this mechanism sensing is kind of dimensionally independent. And we can also look at a few particles as well. We can say, well, what happens if we don't just take 1, we take 2, or if we take k particles? So if we average over the first k particles, what we find is that this is the error here up on the, it gets slightly worse. It's got this term in it, this error term here, which should be 0. So you have to look at this term log k factorial over k and ask, is that increasing or decreasing as a function of k? It is increasing very slightly, but the variance is reducing. So by taking more particles, you're getting a worse and worse estimate. And the basic thing here is that the first particle has the most information, the second has the next most, the third has the next most. So as you take more and more particles into your average, you're getting a worse estimate, but you are reducing variance. For example, taking 5 particles, you get your error increases by 10%, yet your variance goes down by a factor of 5. So this trade-off you can explore. Like, all right, every time I average over more particles, I'm getting a worse estimate, but a lower variance. And we've found that about 10 to 12 is the sort of optimal number of receptor events you want to average over. And that balances a minimization of the error and a decrease in the variance. Okay, so the 2 sort of points here is that those 3 examples here where the sort of time scale of this biological process was governed by extreme statistics. And they're very reliable. You can turn something stochastic into something very reliable. And usually that early information, that hunch you get very quickly, has a lot more quality of information in it than all the stuff later. If you sit and wait and accumulate information, there can be a lot of lower quality estimates that you're building into your average. All right, so thank you again and happy birthday, John.
Thank you. Any question?
Can you go back and detail the inverse problem solution by the cell?
Yeah.
And in passing, you remind me the Dirichlet model